myself. My name is Natalie Caldell. I am the founder and the executive director of BioBuilder Educational Foundation, the nonprofit that has been coordinating this online idea accelerator. It is a program that draws a lot of its inspiration from the teaching that I have been doing at MIT in the Department of Biological Engineering.
Methicillin-resistant Staphylococcus aureus, MRSA, is a huge problem in hospitals. It is very difficult to treat and clean up because it is resistant to some antibiotics. We designed a spray that would make MRSA glow when the spray comes in contact with it, making it easier to find and clean.
So our group has chosen to look at the way to counterbalance the effects of formaldehyde from the human embalming process in attempts to limit the spread of released gases into the ground and waterways.
We decided to start with bacteria known as methanotrophs. Methanotrophs are known for living off of methane gas. With this knowledge, we want to alter the methanotroph to eat formaldehyde.
Hello, this is Group Orange, and welcome to our BioBuilders project showcase.
20% of Americans have chronic pain. This means that over the past 6 months or more, they have been in pain almost daily. To manage this pain, some are prescribed heavy-duty opioids. Because bacteria are reactive to stimuli in environment and can create proteins, they show promise as a method of responsive pain treatment.
For our project, we decided to focus on environmental issues like the abundance of microplastics, due to being from communities that revolve around fisheries as a main economic source. A study in Blue Hill, Maine in 2014 showed that on average, 14 microplastics per liter were derived from the Penobscot Bay.
The microplastics absorber, we will use a constitutive promoter so that it's always on to detect microplastics. For the open reading frame, we will use a gene from the Pseudomonas bacteria. This bacteria absorbs microplastics as energy, so we would want that genetic piece.
Hi, I'm Logan Williams, and I'm a part of the Bacteria Light Bulb Project.
For our project design, we wanted to make a Shigella bacteria that could power a 60-watt LED light bulb. The input would be normal bacteria ATP, output being connected to the circuit.
Solving microplastic pollution through algae. We are the Micro Dragons team from Julia Reynolds Masterman High School. The topic area we decided to work on was environmental science because our team's ideas initially revolved around microplastics, specifically those polluting our oceans.
The system we designed should absorb the ocean's microplastics through a strain of ocean algae, green filamentous algal cells, and then produce a vibrant neon color within the algae to facilitate human cleanup of ocean microparticles.
Hello, we are the Red Team from Masterman High School in Philadelphia, Pennsylvania. The topic area we decided to work on was the environment. We've decided to focus on developing modified plants that would conduct photosynthesis at faster rates to more effectively and rapidly reduce the amount of CO2 in the air.
We are the Yellow Team from Julia R. Masterman High School in Philadelphia, PA. The topic area we decided to work on was medical. We've decided to focus on figuring out a way to bioengineer certain peptides that would bond to the acetylcholine receptors, the same binding sites in which nicotine binds to.